Okay. Okay, so this is the slide. Okay, so I hope you all can see the slide. Okay, so basically for this chapter, we are going to look at the chapter on gene technology. Okay, so boleh dengar ya semua?
Boleh, Madam.
Okay. Okay, so basically dulu kan sebenarnya chapter 2 ni adalah apart from dia adalah sebahagian of chapter 1. So imagine chapter 1 ni sangat besar tapi dia terpaksa bahagikan kepada 2. Okay, so dia jadi chapter yang baru which is this chapter 2. Okay, so chapter 2 is on gene technology. Okay, so basically in order for you to understand this chapter, you have to really understand chapter 1 lah. Okay. Okay, so the learning outcomes of this chapter is to explain the principles of recombinant DNA. Okay, so recombinant DNA technology. So recombinant DNA is when you combine together the DNA from two different species, two different organisms, two different species. Okay, so for example, you insert a gene from gene that you want to produce something, a protein, for example. The protein can be the product can become hormones, for example. Okay, so hormones. So instead of you extracting hormones from animal, okay, you insert sebab kadang-kadang kita boleh meragui, okay, apa hormon yang extract daripada animal ni halal ke tak, kan? Macam insulin untuk treat diabetes, for example. So you extract the insulin from an animal. Okay, so kat situ boleh jadi punca keraguan halal ke tak semua, kan? So through gene technology, you insert the gene of interest that can produce the hormone insulin into bacteria. Okay, into bacteria. So bacteria, inside bacteria dia ada dua jenis DNA. Satu is the DNA in the nucleoid region, the chromosome of the bacteria, and the other one is a small circular DNA, okay, which we call it as plasmid. So plasmid ni banyak digunakan dalam gene technology lah, kan, untuk menghasilkan certain protein product, okay, hormone, enzyme, whatever lah, kan. So kalau awak tengok dekat cheese, for example, kan, so ada enzyme yang cheese tu awak tahu dia adalah susu yang kita ambil dia punya fat, solid fat dalam susu tu, okay, so dipejalkan. So in order to make the cheese solid, you have to insert enzyme to solidify the cheese, kan. So enzyme ni dia selalunya diambil daripada rennet. So rennet tu daripada enzyme rennet, RENNET. Kalau pergi, awak suka makan cheese kan, awak pergi tengok labeling dekat cheese yang kat supermarket tu ada rennet. Okay, so this rennet sama ada dia dihasilkan oleh bakteria ataupun you extracted from the stomach of a baby calf. Okay, so either one kan. So in order to get the rennet is from the production of the enzyme through this gene technology, or you extract from the calf, okay, the stomach from the calf, okay. So that is one of the example for the application of this gene technology, okay. So second, describe details on recombinant markers. So what is recombinant markers? Recombinant markers ni adalah pada plasmid bacteria tu dia ada satu segmen gene, okay. So, dia ada segmen gene yang salah satu contoh yang kita akan tengok dalam chapter ni adalah kita panggil dia sebagai AMPR, ampicillin resistant gene. So gene ni dia akan membolehkan bacteria to survive in certain condition. Okay, in certain condition. So that is, recombinant marker ni adalah sangat berguna dalam kita menentukan sama ada bakteria tu dah ambil tak recombinant plasmid tu ke belum. Okay, so kalau bakteria tu survive dalam medium, contohnya okay, kita nak grow bakteria ni dalam medium yang ada ampicillin. Okay, ampicillin. So kalau bakteria yang tak ambil plasmid, dia akan mati. Okay, tapi kalau bakteria yang ambil recombinant plasmid ni, dia akan able to survive. Okay, so that is the function of recombinant markers. Okay, to identify whether the bacteria has taken up the recombinant plasmid or not. Okay. Number 3, explain the use of DNA library. Okay, so the DNA library adalah satu database yang mana dia bukan library ada macam buku macam kita punya kan. Dia adalah satu database yang ada semua DNA sequence of an organism. Kan, so kalau macam kita DNA library ni kita ada genome library kan. So kita ada cDNA library yang kita akan tengok lah. Okay, so genome library will have all the sequence of a genome of an organism. Genome tu tahu kan? So what is genome? Genome tu is the whole genetic composition of an organism. Genome lah, kita ada, kalau human kita ada 46 chromosome kan. So all the DNA in that 46 chromosome will the sequence, okay, of that DNA from the 64 chromosome will be in the genome library. For human, for example, adalah semua ada, there are genome library for many other organisms. Okay, so it contains all the DNA sequence. Okay, termasuklah coding and non-coding, the introns and also the exon of the DNA. Okay, so tapi kalau another library is cDNA. So what is cDNA? cDNA ni dia hanya akan ada coding sequence sahaja. So information daripada cDNA lagi reliable lah kan, sebab dia ada all the coding sequence only. Okay, it has the exact gene, okay, that can produce certain product, protein product for that gene. Okay, so explain the use of cDNA. cDNA, C is stand for complementary DNA. Okay, complementary DNA is the DNA that is produced, okay, using mRNA as template. Kita belajar kan, so mRNA it only contains, for eukaryotes, it only contains the coding sequence. Okay, so when it only contains the coding sequence, from that mRNA you use it as a template to synthesize DNA molecules that only contain the coding sequence. Okay, so daripada situ awak boleh insert the gene of interest into the bacteria, okay, into the bacterial plasmid. So from then, for sure, the bacterial plasmid can produce the protein products, okay, the enzyme or the hormones. So number 5, discuss the application of recombinant DNA technology. So banyaklah application dia kan, dalam medicine, dalam agriculture, okay, to the environment, okay, gene therapy. So there are many applications of recombinant DNA technology to produce modified organism, okay, GMO. Okay, tapi kena ada etika dia lah, alright. And then number 6, describe the gel electrophoresis technique to separate and visualize DNA fragments. Okay, so DNA fragments that you extract out, jumlah fragment dia, jumlah DNA obviously sangat sikit. Okay, jumlah DNA adalah sangat sikit. So you use a technique which is dia ada dua teknik yang berturutan. Okay, so satu adalah PCR kan, polymerase chain reaction. So this is to amplify the amount of DNA that you extracted from the organism. Okay, so the amount of DNA will not be enough. Okay, sebab dia dalam kuantiti yang sikit. So in order to amplify the quantity of DNA, you use PCR, polymerase chain reaction. Okay, a machine that can amplify the amount of DNA. So after you have amplified the amount of DNA, then you insert DNA into a gel and you run for gel. Okay, so gel electrophoresis is to separate DNA fragment into separate centrifuge the DNA dalam sistem chloride. So DNA with the densest density akan pergi kat bawah, the lightest density akan ada kat atas tube tu. Gel electrophoresis lebih kurang sama. You akan you will spin, okay, centrifuge simple here.
Sorry, ada orang ask untuk masuk tadi.
Oh ya, kejap. Okay. Kalau ada awak dengar ada suara baby ni, so maksudnya saya tengah pangku anak saya ni sambil mengajar. So excuse the extra noise. Okay. Quiet. Alright. Okay. So gel electrophoresis. So nanti kita akan tengoklah teknik dia untuk gel electrophoresis tu. Okay. Okay, again. Alright. So recombinant DNA technology. So principles and method. So lagi nama saya. Okay. Okay, so recombinant DNA technology, principles and methods. So you have to know what the definition for DNA technology. So it is a technique for sequencing and manipulating DNA. Okay, sequencing, it means that the DNA that you have extracted from an organism, you run for gel electrophoresis and you get the DNA, and then you send the DNA sample to a company, and then the company tu akan determine the DNA sequence, okay, for that gene. Okay, kita tak boleh kita dengan awak cara extract je and then you spin and then you go for gel electrophoresis, will not be enough for you to determine the sequence. Sebab DNA sequence is, you should know the bases kan, so the sequence of bases for that DNA. So DNA sequencing selalunya kita akan hantar kepada satu company ni, so company tu akan determine, okay, what are the sequence for that gene, okay. And after you have determined the DNA sequence, then you can use the information to manipulate that information kan. So nak buat nak buat cDNA ke whatever lah kan. So nak insert the gene into plasmid, and then you insert into bacteria. Okay. So that is gene technology. Okay. So the next one is recombinant DNA. So recombinant DNA is DNA molecule made in vitro, in test tube, okay, inside lab, okay, inside lab. You produce recombinant DNA using in vitro, okay, using test tube in the lab with segment from different sources, from different organisms, okay. You insert gene from human, gene of interest, for example, and then you insert into bacteria in order for that bacteria to produce the protein product. Okay, the protein product can be enzyme or hormones. Okay, so gene cloning is a process to produce multiple copies of specific DNA segment. So sebab tu kita insertkan gene of interest dalam plasmid of bacteria, sebab as you know, bacteria can multiply really fast. Okay, so when the bacteria divide for so many generations, so it can produce many copies of the gene of interest. Okay, so when bacteria produce many copies of gene of interest, you get the product produced, ataupun you can get the gene of interest to insert into another organism. Okay, kejap ya. Excuse me, saya nak hantar anak saya ni ke husband saya dah. Okay, so let's continue. Sorry for the interruption. Okay, so gene cloning is a process to produce multiple copies of a single gene, and two basic purpose of gene cloning is to make many copies, to amplify, okay, of a particular gene. How to amplify the gene is through the division of the bacterial cell. Okay, so and to produce a protein product. So once the bacterial cell has divided so many times, so obviously it copies also the gene of interest and produce more of the protein products, okay, enzyme or hormones. that you desire. Okay. 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 Okay, so 6 tools used in recombinant DNA technology. Okay, so you have to have uh, the substance lah kan, tools, okay, used to produce the recombinant DNA and to apply the recombinant DNA into whatever you want, okay. Whether to extract the protein product or to get the gene of interest and insert into another organism, okay. That is to produce GMO, okay, a genetically modified organism, something like that, right? Okay, so target DNA. So target DNA is the DNA that you target that has the gene of interest that code for the production of certain protein products, okay, enzyme or hormones, okay. So that one kena determine lah nak ambil daripada organism apa, kan, manusia ke haiwan ke, so you have to determine. Okay, so contain gene of interest such as gene responsible for insulin production, okay. So and then extracted from organisms such as human cell. So obviously when you want the product such as insulin, you have to extract out the gene, okay, from that from the cell that produces the insulin, okay. So in this case yang awak nak adalah insulin kan, so you know that insulin is produced by what organ? Insulin dihasilkan oleh organ apa?
Pancreas.
Pancreas kan. Okay, so you get the information by extracting out the DNA sample from pancreatic cells. Okay, so the gene you cut using restriction enzyme, kan, and then you insert into the plasmid. Okay. Okay, so this is restriction enzyme. So it is also known as restriction endonucleases. So awak boleh gunalah mana dua nama ni sama je, restriction enzyme or restriction endonucleases. So used in gene cloning and genetic engineering, found naturally in bacteria. So name for the bacteria from which they are isolated. So there are many restriction enzymes, okay. So such as for example EcoR1, kan. So EcoR1 is the restriction enzyme extracted from E. coli, kan, E. coli, bacteria cell. So this enzyme will recognize and cleave is to cut, okay, cut at restriction site yang spesifik. Okay, so kalau untuk EcoR1, dia hanya akan mencari dan cut, it will only find and cut restriction site that has this sequence GAATTC, kan, GAATTC. So bila awak tengok sequence pada kedua-dua DNA strand ni, dia adalah sama for in both direction. Kalau tengok strand atas, dia baca GAATTC, bawah pun GAATTC. So this is what we call it as palindromic sequence. Palindromic sequence maksudnya bacaan strand atas dengan strand bawah sama. Okay, in opposite direction, they are the same. So salah satu ciri restriction site ni is that they are palindromic sequence, which is both strand will read the same in opposite direction. Okay, so nature of cut ada dua jenis, which is sticky ends and also blunt end. Sticky end tu maksudnya apa? Dia cut in a staggered manner. So ini dia akan produce sticky end. Bila restriction enzyme which is EcoR1 cut in between G and A, so these two strands will separate, produce a gap. Okay, so it cut here. So this fragment and this fragment will separate out. Okay, it will separate. Okay, so it produce two ends that are not that has the two sticky ends, and then you can insert, okay, the gene of interest in between here. Okay, so in order to insert the gene of interest, you also have to cut the DNA that contains the gene of interest using the same restriction enzyme. Why do you need to cut using this the same restriction enzyme? So that it can produce the same sticky ends that are that can complement to each other when you insert into the bacterial plasmid. Okay, so ingat you have to cut the bacterial plasmid and also the gene of interest using the same restriction enzyme so that it can produce the same sticky ends. So when you insert into when you insert the gene of interest into the plasmid, they complement each other. Okay, they complement each other. So selalunya kalau dalam exam dia akan guna yang common enzyme inilah EcoR1. So at least you have to know the sequence of this restriction enzyme for EcoR1. Okay, it produces sticky ends. So another enzyme is HIN3. So this is extracted from the bacteria Haemophilus influenza. Okay, so this HIN3 will also cut in a staggered manner and produces sticky end. Okay, sticky end. So it cut between A and A here and A here. So as you can see, the sequence for the two DNA strands are the same in opposite direction. So they are palindromic sequence. Okay, palindromic sequence. So BAM HIN1. So from this bacteria, Bacillus amyloquifactions. Okay, so so this cut in a staggered manner as well. Okay, so produces sticky ends. And then SMA1 from this bacteria. So this one cut straight. So it cuts straight and produces blunt end. Okay, so yang tu kena tahulah kan. So bila dia cut straight, it produces blunt end. So ada dua jenis cut, dua jenis end yang akan dihasilkan. Either it cut in a staggered manner, produces sticky end, or cut straight, produces blunt end. Okay. Okay, so this is the diagram to explain. Okay, so this is the bacterial plasmid, for example. Okay, so the bacterial plasmid is DNA, double-stranded DNA molecule. And then the restriction enzyme will recognize restriction site. So this is restriction site. Okay, that has palindromic sequence. Okay, palindromic sequence. The top strand and the bottom strand has the same sequence in opposite direction. Okay, so in this case, the enzyme used is HIN3. So it cut in a staggered manner. Okay, and produces sticky end. Okay, sticky end. Inilah macam ni. Dalam exam dulu pernah keluar soalan yang mana dia okay. Selalunya soalan ni dia tak adalah kejam kan. So dia dah bagi contohnya dia dah show kat sini kan. So ini soalan yang lepas lah. So you just have to produce and draw balik the sticky end produced. Can sticky end produced. So you have to make it clear that the two DNA strands separate out. Okay, and you show it clearly that it produces sticky end here. Okay, ni kan mesti ada gap lah kat sini kan. So this is the gap where you insert the gene of interest. Okay, so this is where you insert the gene of interest. So the gene of interest you cut with the same restriction enzyme. Okay, so another example is you cut using EcoR1 kan. So the sequence is GAA-TTC. So it cut in a staggered manner, produces sticky ends. Okay, make sure when you produce the sticky ends, never forget to write the ends, these two direction here. So bila atas 5 kat sini, obviously hujung kat sini 3 kan. So bawah dia akan complement dengan 5, hujung ni dengan 3. Okay, so make sure never to forget to write the two ends here. Okay. So kalau dah penat-penat kan draw ni dah cantik dah produce sticky ends, but you did not label the two ends here, then jawapan awak akan ada tendency untuk salah lah. Okay. So this one SMA1 Okay, digest. So the enzyme cut in a straight manner produces plant N. So this is the region where you insert the gene of interest. Okay. Okay, so restriction enzyme are very specific. So every restriction enzyme you extract from different bacteria will recognize the restriction site. Okay, what is the characteristics of the restriction site? They are palindromic. What is palindromic? The sequence is identical when read in the opposite direction in the complementary strand. So these two DNA strands, they have the same DNA sequence that read in the opposite direction. GAA-TTC, bawah pun GAA-TTC. Itu yang kita namakan ni sebagai palindromic. Okay. So cut or cleave. So kalau awak boleh guna dua-dua istilah. Okay, untuk explain macam mana cara restriction enzyme ni berfungsi. It cut or cleave, either of the two words. Okay, kalau nak canggih, guna cleave lah. Kan, kalau biasa-biasa cut lah. Okay, semua mana betul. Okay, so cut or cleave double-stranded DNA molecules at specific nucleotide sequence, which is the restriction site. So this is called as restriction site. Okay, the site where the restriction enzyme will cut. So another feature of restriction site is that the length is short, which is about 4 to 8 nucleotide long. Okay, nucleotides long. So kalau GATC ni ada berapa? Ada 6. Okay, ada 6. So dia punya length dia is not too long lah. Lagi pendek daripada primer. Alright. So make many cuts in the DNA molecules, producing a set of restriction fragments. So this is because any sequence this short usually occurs many times by chances along DNA molecule. Kita adalah eukaryotes. Kan, contoh kita adalah eukaryotes. Our DNA is really large. Okay, it's really large. So the chance of restriction enzyme encountering this sequence many times is high. Kan? So when you extract DNA from an organism and you insert the restriction enzyme into the reaction, the enzyme will cut the DNA into many fragments. Contohnya kat sini. Okay, so this is a cell. And inside the cell you have the DNA. Okay, the DNA. So you extract out the DNA and then you insert into a test tube, for example. And so DNA, and then you insert restriction enzyme. Then so the restriction enzyme will cut the DNA into many. Okay, and when it cut the DNA into many fragments, bukan semua cut ni akan ada gene of interest. So maybe ni je akan ada gene of interest, for example. Okay, so that's why you need to do DNA sequencing. Okay? And know the exact sequence for the gene that can produce that certain protein. Okay. Okay, so next. Okay, so kita ada apa tadi yang first adalah, okay, you have to have the target DNA. So in order for you to get the target DNA, you have to know the sequence of the gene. Okay. And then you need a restriction enzyme. Okay. And then next is DNA ligase. Okay. Kita dah belajar kan itu masa DNA replication. The function of DNA ligase is to attach together the Okazaki fragment, to join the Okazaki fragment into continuous lagging strand. Okay, so the function of DNA ligase is to join fragments of DNA. So in this case, you want to use DNA ligase so that you can join the plasmid DNA plasmid of bacteria. Okay, with the gene of interest. So this is gene of interest that you extract out from a certain organism cells. Okay. So this is an organism cells that you extract out the gene and then you insert into the plasmid. So kat sini you have to join together the gene of interest with the plasmid, so you have to use DNA ligase. Okay, so DNA ligase catalyzes the joining of DNA from two different sources, okay, which is the plasmid and also the gene of interest. Okay, so when you attach together, when you join together the plasmid with the gene of interest, bila dah bersambung, then you produce recombinant DNA. Okay, recombinant DNA. So, so this is the basic reaction lah. So how the DNA like join the two DNA recall like macam mana you peptide chain. So the bond between the nucleotides are phosphodiester bond. Kan? So you use DNA ligase so that the DNA ligase can catalyze the formation of phosphodiester bond between the nucleotides. Okay, between the phosphate and the sugar of the subsequent nucleotides. Kan, ini saya draw balik untuk awak recall lah. So this is the first nucleotide, okay, for the chain. So katakanlah hijau tu ada nucleotide yang last untuk gene of interest. So you want to use the DNA ligase to catalyze the formation of phosphodiester bond between the subsequent nucleotide here. Okay, so this is the phosphodiester bond formed. Okay, formed between the two nucleotides. Okay, so next, okay, after you produce DNA, okay, the plasmid with the inserted gene of interest, then you need to insert the plasmid into a host cell, okay, which is a vector. So cloning vector here, tadi plasmid sama je, okay. So cloning vector is yang paling common adalah bacterial plasmid. Okay, the most common is bacterial plasmid. So widely used in gene technology, okay, or you can use cosmid. So you can read up on what cosmid are, or bacteriophage ataupun yeast artificial chromosome. So yang kita akan concentrate untuk kita punya syllabus adalah bacterial plasmid, okay, sebab it is commonly used. Okay, so what are the characteristics of bacterial plasmid? So number one, it is a small double-stranded circular DNA molecule that replicates separately from the bacterial chromosome. So this is a bacteria, for example, E. coli. So this is the chromosome of the bacteria in the nucleoid region. Okay, and then you have a small circular DNA. Okay, a small circular DNA. Okay, here, over here, for example. So this is the DNA plasmid of that bacteria. So in order for you to do your to produce your recombinant plasmid, you have to extract out the plasmid from the bacteria. So sekarang ni bila dah keluarkan plasmid daripada bacteria, okay. So this is the bacteria with into a test tube that contains the bacterial plasmid. Masukkan dia dalam test tube, alright? Into a tube, alright. So number two, the second characteristics of bacterial plasmid is that they have dominant selectable marker. Okay, dominant selectable marker to detect recombinant DNA for selection, such as, for example, ampicillin resistant gene, which ampicillin resistant gene, AMP to ampicillin, R to resistant. Okay, ampicillin resistant gene. So when this gene basically, okay, it is inside the plasmid. Okay, it is inside the plasmid here. So this is, for example, ampicillin resistant gene. Okay, so this gene allows the bacteria to be able to survive in certain conditions, such as, for example, because this is ampicillin resistant gene, so when the bacteria has the plasmid that has the ampicillin resistant gene, it allows the bacteria to survive in a medium that contains ampicillin. Ampicillin is antibiotics, kan? Kalau bacteria tu has no plasmid, when it has no plasmid, so when you grow the bacteria inside a petri dish that has ampicillin, so this will cause the bacteria to die. Okay, to die. But when you grow this bacteria in a medium that contains ampicillin, so this bacteria will able to survive. Okay, so because it has the plasmid that has the ampicillin resistant gene. Alright. So number 3, have a unique restriction enzyme cleavage site. Okay, for the insertion of DNA sequence that are to be cloned. Okay, so restriction site ni pada plasmid tu dia akan located at another region. For example here. So selalunya restriction site ni dia akan ada in between another gene. And so this is another gene here, for a segment of another gene that is capable of producing a certain enzyme yang lain juga. So gene tu, region yang mana adanya restriction restriction site, kita namakan dia sebagai reporter gene. Reporter genes. So selalunya restriction site ada dalam reporter genes. Okay, restriction site ada dalam reporter genes. Nanti kita akan tengok what is the significance of having this restriction site inside the reporter gene. Okay, kalau dalam E. coli ni dia akan ada like Z gene. Kita belajar hari tu kan like operon, right? Okay, so like operon, dalam like operon sequence ada restriction site of enzyme EcoR1. So EcoR1 bila dia cut like Z gene ni, the like Z gene will not be functional anymore and cannot produce the enzyme beta-galactosidase. Ingat hari kita belajar like operon, like Z gene will produce beta-galactosidase to hydrolyze breakdown lactose into glucose and galactose. Tapi bila awak cut, okay, so this is like Z gene. Like Z gene kan. So inside the like Z gene you have the restriction site. So when you cut, okay, the gene at the restriction site here, so now the like Z gene cannot function. Okay, it is not functional anymore. It cannot produce the enzyme beta-galactosidase. It is not functional anymore. So the significance of reporter genes ni adalah untuk scan mana bakteria yang dah ambil recombinant plasmid, mana yang bakteria yang tak ambil recombinant plasmid. Okay, so ada dua. So satu adalah selectable marker yang ampicillin resistant gene, satu lagi adalah reporter gene. So saya draw balik, ini adalah plasmid, satu region yang dia akan ada selectable marker such as ampicillin resistant gene, satu lagi dia akan ada restriction site dalam reporter gene. Okay, dalam reporter gene. Alright. Okay, so how recombinant plasmid or DNA are formed? Okay, so this is bacterial plasmid. Remember, bacterial plasmid are double-stranded DNA. Okay, double-stranded DNA, circular double-stranded DNA that are found in bacterial cell. Okay, so here you have the restriction site. For example, GAA-TTC. Okay, GAA-TTC. So you cut the restriction site using a restriction enzyme. Okay, cut the restriction site using restriction enzyme at the sugar phosphate backbone in the 2 DNA strand in a staggered manner. Okay, because the site is GATTC, so it is cut by EcoR1 in a staggered manner, producing sticky ends. Okay, So this produce double-stranded restriction fragments with sticky ends. Okay, so next, so this is the plasmid that you have cut, can. So you have gap between the plasmid. Alright. So ini dah cut. Okay, the plasmid dia dah terbuka, double stranded lah saya draw dia. Kan. So satu panjang, satu pendek. So in a staggered manner. So here you have the gap in between the cut. So here you insert the gene of interest that you have cut using the same enzyme. So it produces the same sticky ends, okay, that complements with the sticky end of the plasmid. They complement each other, okay, because they are cut by the same enzyme. They are cut by the same restriction enzyme. Okay, so this is the DNA fragment that you extracted out from an organism that contains gene of interest. It has the gene of interest, okay, from other organism, from other species. Okay, it has the gene of interest, such as, for example, the gene that can produce insulin. So other species, this can be human. Okay, so this is the bacterial plasmid. Okay. So base pairing of sticky ends, short single-stranded extension. Okay, this one here and this one here. Okay, with complementary sticky ends on other DNA fragments, which is here. Okay, DNA that has the gene of interest, producing various combinations. So fragment from different DNA molecule cut by the same restriction enzyme. So ingat ya, tak semestinya DNA fragment contains the gene of interest. It has other DNA fragment yang kebetulannya dia mempunyai sticky end yang sama sebab dia di-cut oleh enzim yang sama. Sebab kita punya DNA sangat besar. Okay, so the probability of the restriction enzyme to cut and produce many DNA fragments are high lah. So it produce many DNA fragments that may not contain the gene of interest. Okay. So this is one possible combination. So region ni sama ada the gene of interest ataupun tak ada gene of interest. Okay. Due to the many fragments that can be produced. Okay, so after you have inserted the foreign DNA, so this is the foreign DNA, okay, foreign DNA, this is the plasmid. So after you have inserted the foreign DNA into the bacterial plasmid, so you have to join together the two DNA sources, okay, using the enzyme DNA ligase. Okay, so DNA ligase catalyzes the joining of DNA from two different sources, producing a stable recombinant DNA molecule. Okay, so this is the recombinant DNA recombinant plasmid. So ada bila contohnya awak ada banyak bacterial cells. Okay, you have made so many bacterial cells. Okay, so this is the nucleoids of the bacterial cells. And then this is one test tube. Okay, so this you have the bacterial cell that you have remove the plasmid. You have removed the plasmid. So lagi satu test tube ni akan ada plasmid yang dah cut dengan restriction enzyme. Okay. And then you insert also DNA fragment from the organism. Okay. So, yang purple ni DNA fragment from other species. So this is the reaction, kan? Reaction yang berlaku bila awak insertkan foreign DNA and plasmid into one test tube. So probability dia adalah satu. Kalau awak tengok plasmid ni dia akan sama ada boleh ambil foreign DNA fragment ataupun dia tak ambil langsung, dia cantum balik. Kan, dia cantum balik. So this is one possibility. Plasmid join back together without taking any foreign DNA. Ataupun Dia akan ambil the foreign DNA. Okay, foreign DNA. Macam tu. So those are the possibilities. So ada plasmid yang ambil foreign DNA, ada plasmid yang dicantum balik semula. Okay, macam tu. Okay, itu gunanya like Z gene. Okay, like Z gene is to screen whether a bacterial cell has taken recombinant plasmid ataupun plasmid without any inserted foreign DNA. Faham ke? Macam ni lah. Yang ni nanti kan awak masukkan dalam bakteria yang dah tak ada plasmid. So, bakteria ni sama ada satu possibility dia are bacteria that did not take any plasmid. Ingat, tengok ni. Ni adalah bakteria that did not take any plasmid. Yang ini bakteria yang ambil recombinant plasmid. Sekejap. So dia are bacteria that did not take any recombinant plasmid. Dia ambil plasmid yang dah cantum balik semula. Dia ambil plasmid yang cantum balik semula kosong macam tu. Okay. So ada 3 possibility. Bacteria that did not take any plasmid, bacteria that take up recombinant plasmid, bacteria that take up the plasmid without the gene of interest ataupun foreign DNA. Ini. So there are 3 possible outcome tu nanti bila awak buat gene cloning tu. Alright? Itu kegunaannya yang macam mana nak tahu bakteria ambil plasmid ke tak, itu kegunaan yang marker gene tu tadi kan. So the marker gene is to screen the bacteria, to screen the bacteria whether the bacteria has taken plasmid or not, whether the bacteria has taken recombinant plasmid or not. That is the function of the next tool, which is the yang tool yang ke-6 ni, selectable marker ni, is for screening. It is to screen the bacteria. So after you have produced the recombinant plasmid here kan, so you need the host cell. So the host cell yang ni lah. So tool yang ke-5 adalah the host cell, yang bacterial cell. Okay. Ibu. Ain, dia kena buat kelas lain. Can you go outside please? Saya tapi tak jumpa. Mana ada dekat luar? Mana tu ada dalam ni? Tak nak pegang. Keluar. Okay, so this is the host cell. So host cell is an organism or a cell to produce recombinant DNA. So characteristics of host cell. One, it can allow replication of recombinant DNA. Okay, so selalunya kita guna bacteria cell ataupun virus. So we use also. Okay. So able to accept recombinant DNA via transformation. Bila host cell tu dah accept the recombinant plasmid, proses tu kita namakan dia sebagai transformation. Okay, when a host cell accepted the recombinant plasmid, the process is called transformation. It is transformed. And then multiply the gene product of the recombinant DNA. Bila bacteria tu multiply, so it also multiply the recombinant DNA. Ya. Okay. So the next characteristics is that able to maintain structure of recombinant DNA from one generation to another. So kita, kalau bacteria cell ni dia akan maintain lah kan the sequence of the recombinant DNA without changing the sequence, okay, of the recombinant DNA as the bacterial cell multiply or divide. Okay. So these are the 4 characteristics of the host cell. Okay. Number 1, can allow replication of recombinant DNA. Masa bacteria tu multiply, dia akan juga multiplykan recombinant DNA. Able to accept recombinant DNA through transformation. And then multiply the gene products. Okay, gene product tu is the enzyme lah. So bila bacteria cell tu membahagi, dia juga akan menghasilkan gene product tu juga. Kan, dia boleh, kalau ada gene of interest yang boleh hasilkan insulin, so maksudnya sekarang ni bakteria tu boleh dah menghasilkan insulin juga. Okay, so able to maintain structure of DNA, recombinant DNA from one generation to the other. Okay, so example of host cell adalah E. coli lah kan, bacteria cell yang commonly used, and also plant cell. Okay. Okay, so the last tools that you require adalah marker genes. Okay, so these are marker genes. So marker genes ni terbahagi kepada 2. It is divided into 2. One is selectable marker and the second one is reporter gene. Okay, selectable markers and reporter genes. What are selectable markers? Genes with which enable selection of transform cells in growth media containing selecting agents such as antibiotics. So example of selectable markers are ampicillin resistant gene and TETR gene, which is tetracycline resistant gene. Okay, so ini ada pada plasmid tu. So by bacteria having plasmid that has the selectable marker ampicillin gene, it allows the bacteria to be able to survive in the medium that contains ampicillin. Kalau bacteria tu tak ambil plasmid, maksudnya dia akan mati kan, sebab dia tak ada ampicillin resistant gene. Faham So second adalah reporter genes. So encodes for product that can be easily scored or quantified. This is to detect or screen transformed products. Okay, two examples of reporter gene is like Z gene, and the other one is a green fluorescent protein. Okay, like Z gene ni contohnya pada plasmid bacteria tu lah kan. So dia ada like Z gene which is to produce enzyme beta galactosidase for blue white screening. So kat sini kita akan tengok lah kat bakteria yang ambil recombinant plasmid, dia akan hasilkan colonies yang dia akan hasilkan 2 colonies, sama ada koloni biru ataupun koloni warna putih, white colonies ataupun blue colonies. Okay, so you have to select which one of the bacterial colony that contains the recombinant plasmid. Okay, the other one is gene fluorescence protein. Make cells glow under UV light using fluorescence microscope. Okay, ini contohnya lah green fluorescent protein GFP. GFP ni dia adalah protein yang dihasilkan oleh jellyfish that allows the jellyfish to glow lah kan, when exposed to blue to violet ultraviolet UV light. Okay, the GFP protein glows bright green kan, so it allows the jellyfish to glow okay under UV light. So this gene from the jellyfish kan, gene daripada jellyfish ni yang boleh hasilkan protein that allows the jellyfish to glow ni is inserted into the bacterial plasmid. So dia akan insert the bacterial plasmid kat sini. Green fluorescent protein gene. Yang ini adalah GFP, gene yang di-extract daripada jellyfish masukkan dalam plasmid bacteria. Okay, plasmid bacteria. So kalau inilah to insert into the bacterial gene kat sini. So bila bakteria plasmid dah ada GFP protein ni, GFP gene ni, dia boleh menghasilkan protein yang membolehkan bakteria tu glow juga. Okay, so when you insert the GFP gene into the bacterial plasmid, now the bacteria can also glow under UV light because now the bacteria is capable of producing the green fluorescent protein that was initially produced by the Okay, so the process of gene cloning, kita break dulu sekejap. Okay, you all tengok balik the 6 tools, fahamkan, kan kita break 5 minit. Kalau ada soalan boleh tanya. Ada soalan? So far untuk the 6 tools for gene cloning. Fahamkan dulu baru kita boleh proceed kepada the next component of this topic, which is gene cloning. Okay, tengok balik tools dia. Target gene, restriction enzyme. Restriction enzyme, it cuts the plasmid and also the DNA fragment from other organisms, okay, that has the gene of interest. And then you have to have the vector. The vector usually is plasmid. And then you have to have DNA ligase to join together the foreign DNA with the plasmid to produce recombinant plasmid. And then you have to have the host. All boleh sign attendance dulu. Okay. Okay, so we are now going to proceed with the next section, okay, which is gene cloning. Okay, the process of gene cloning. So gene cloning using gene cloning using bacterial plasmid as vector. Vector is the plasmid lah, kan? Vector is the plasmid, which is vector is actually the DNA that is capable of carrying the gene of interest. Okay, so that is vector. So gene cloning using bacterial plasmid as vector. So the first stage, isolation of target gene. Okay, so extraction of plasmid from bacterial cell and gene of interest from different sources such as prokaryotic or eukaryotic cell. So you have to extract out the plasmid from bacterial cell, foreign DNA from another species that contains the gene of interest. Okay, region of interest, gene of interest that can produce certain protein products, such as, for example, certain enzyme or hormones. Okay, so that is the first step that you have to do. Isolation. So yang first ni, yang untuk ini kan, dia asalnya daripada E. coli. Yang saya draw balik, so dia ada nucleoid region that has the chromosome of the bacteria in the nucleoid region and also the plasmid. Okay, so what you do is that you extract out the plasmid from the bacteria, you insert into a test tube, into a tube. So this is isolation of plasmid. You isolate. So now the bacteria, when you have extract out the plasmid, the bacteria now has no plasmid. Kan? It has no plasmid anymore. Dia ada DNA at the nucleoid region only. Okay? Macam tu. Alright. So the foreign DNA, you have to extract out the foreign DNA from an organism. So contohnya, awak nak, you want the gene that can produce insulin. So awak extract out the DNA from pancreatic cell, for example. Okay, so pancreatic cell, you extract out the DNA from pancreatic cell. Okay, so you extract out and put it in a test tube. So this is the DNA that contains gene of interest. Okay. Boleh nampak? So dalam plasmid ada gene for antibiotic resistance such as ampicillin resistant gene. So asalnya bila bakteria ni dia ada plasmid, dia bolehlah survive dalam ampicillin resistant gene. Okay. So next. So insertion of target gene into a cloning vector. So cloning vectors such as bacterial plasmid has marker gene which is ampicillin resistant gene. Plasmid are mixed with fragment of target gene to form recombinant DNA. Sebelum kita proceed kepada stage 2, dekat stage 1 ni, okay, you have to mix restriction enzyme. Okay, such as, for example, eco R1. You mix together the plasmid with eco R1. Yang ni pun sama. You mix together the DNA with eco R1. Contohnya, you mix with restriction enzyme. So bila you mix the plasmid with restriction enzyme, okay, yang ni, dia punya produk adalah plasmid tu akan terbuka lah. Faham? Okay, plasmid tu akan terbuka. Macam ni. Alright. Plasmid terbuka because now you have cut the plasmid. Okay, sorry. Tadi saya sampai mana tadi? Still stage 1 ke? Kita sampai stage 2.
Ya, awak.
Stage 1, madam.
Saya explain balik semula ke stage 1 daripada awal? Sampai mana? Saya explain.
Madam ada cakap yang kena letak restriction
Campur.
enzyme dalam kod tu. Ya,
Sampai situ je. Okay. Okay, so kita tengok balik stage 1. Okay. So stage 1 as you see here. Okay. So this is the bacterial cell. Okay, in the bacterial cell you have the DNA of the bacteria in the nuclear region, and it also has the plasmid. Plasmid yang warna purple ni. So you have to extract out the plasmid from that bacteria and put it in a test tube. Okay, so this is the test tube containing plasmid from the bacteria, and so now you are left with bacteria that has no plasmid. Okay, so for example, when
you're not presenting.
oh, sorry. So tengok ODL ni bukan student je rasa sangat mencabar, pensyarah pun rasa sangat dicabar kesabaran dia. Kita putus je. Okay. Alright. Okay, so we start again for stage 1. Okay, so kita recap balik Alright, so isolation of target gene. There are two things that you need to isolate. One is the plasmid from bacteria. Second is the DNA from gene of interest. Okay, DNA from species. Okay, other organism that contains gene of interest. Okay, so this is the bacteria. So here, for example, E. coli. So here is the DNA that is within the nuclear region, and you have here the plasmid. Okay, so plasmid daripada bakteria ni awak keluarkan, extract, isolate. When you have isolated, you insert the plasmid into a test tube. Now you are left with bacteria that has no plasmid. Yang ni awak simpan dulu, yang ni awak akan guna nanti kejap lagi. Kan, you are going to use this later. Alright. Okay. So gene for antibiotic resistance. So plasmid will contain gene for selectable marker. Kan? So example of selectable marker is antibiotic resistance, such as ampicillin resistant gene. Okay, so the test tube that contains the plasmid that you have extract and isolate from the bacteria, you mix with restriction enzyme, okay, which is EcoR1. Ini awak take note ya, sebab dalam slide ni tak tulis. So EcoR1. So when you mixed the plasmid with EcoR1, so the function of restriction enzyme is to cut open the plasmid at restriction site. So now the plasmid is opened. Kan dia dah terbuka kat sini. Okay, so that is for plasmid. For the foreign DNA, okay, you extract out DNA from a species that contains gene of interest that produce certain product that you are interested in, such as, for example, insulin. Okay, so so sekarang ni untuk yang senang kan, you extract out pancreatic cells. Okay, so pancreatic cells, you extract out the DNA. Okay, so you extract out the DNA into a test tube. Now you mixed with the DNA with the same restriction enzyme. Okay, kenapa? Sebab nak produce the same sticky ends. Alright. Okay, so you cut the foreign DNA using the same enzyme, and then now you produce many DNA fragments. Okay, so remember, not all the DNA fragment contains gene of interest, but kita punya DNA sangat besar. Okay, so not all. Okay, so maybe this one contains the gene of interest, for example. Alright. So okay, so you cut using restriction enzyme, the same restriction enzyme. Okay, so sekarang ni after you have produced those two test tube, so now what you need to do is that you mix together the DNA fragment from this species that you extract out, you mix into the bacterial plasmid. Kan, you masukkan dia sama-sama dalam satu test tube. Okay, so when you mix together the foreign DNA into the plasmid here, so dia akan jadi yang start yang kedua, which is the second stage. Okay, insertion of target gene into a cloning vector. So when you mix together the foreign DNA and plasmid into the same test tube, it produces recombinant DNA. Okay, so cloning vector such as bacterial plasmid has marker which is ampicillin resistant gene. So plasmid are mixed with fragment of target gene to form recombinant DNA. Okay, so kalau saya draw kat sini, sekejap ya. Okay. Saya buat sekejap. Okay, so ini contohnya. Dalam test tube ni, yang biru ni adalah plasmid kan plasmid. Okay, yang merah ni adalah foreign DNA. Okay, so you mix together. The plasmid, also the foreign DNA together. So the punya outcome dia okay. So akan ada plasmid yang cantum balik semula. So bila cantum balik semula, this is one possible outcome adalah dia akan produce non-recom another possible outcome adalah dia akan produce Allah jap. Draw balik macam tu. Bal pula. Okay. So it has many plasmid and the foreign DNA. Kenapa dia hilang? Kejap ya. Okay. Okay, so one, you will produce non-recombinant plasmid. Okay, plasmid itu disambung balik semula without taking any foreign DNA. So it produces non-recombinant plasmid. So another possibility is that it produces recombinant plasmid. Okay, recombinant plasmid. So ada dua possibility. Okay. Plasmid macam ni. Okay. So dia ada dua possibility. Okay. So ingat tu je. So recombinant plasmid awak boleh assume dia may contain gene of interest and it may not contain gene of interest. Bukan semua DNA fragment ada gene of interest. Okay. So let's say the red fragment here, as you can see from the diagram, and is the plasmid. The red is the foreign DNA and the blue is the plasmid. Okay, so they will produce the same sticky ends that are complementary with each other. So this will produce recombinant DNA. Okay, so possibility dia bila mixkan together sama ada dia produce non-recombinant plasmid. The other one is recombinant plasmid. Sekejap, saya nak keluar balik dia macam dah jadi ni, saya klik PowerPoint ni. Okay. Okay, so stage yang ketiga, introduction of recombinant DNA into host cell, which is transformation. So recombinant DNA is transferred into host cells such as E. coli in a medium containing calcium chloride. Okay, so host cell will reproduce and some of them will take up recombinant DNA. So tengok tadi, kan? Tadi awak dah. Okay, so ini ada medium, kan? Ada petri dish contohnya yang ada bakteria. Okay, that you have removed the plasmid. Okay, that you have removed the plasmid. Okay, so this is the bacteria that has no plasmid. So tadi awak dah hasilkan dalam test tube tadi. Saya draw besar-besar supaya jelas. Plasmid. Plasmid. Okay. Yang bergabung dengan foreign DNA, kita panggil dia sebagai produk yang kedua. Is recombinant plasmid. Sekarang dia pindahkan dia dalam campurkan dia dengan bacterial cell ni. Okay, so campurkan dengan bacterial cell ni. So bacterial cell ni sama ada dia punya outcome dia okay. So dia ada that does not take any plasmid. Okay, ini adalah satu possibility outcome yang akan hasil. Does not take any plasmid. Yang kedua, okay, adalah bakteria. Okay, bakteria that take up non-recombinant plasmid. Contohnya yang ini. Okay, dengan yang ini. Okay, so outcome yang ketiga adalah bakteria that take up recombinant plasmid. Okay, so contohnya adalah bakteria yang ini. Okay, nampak Okay, so ada 3 possibility yang akan terjadi pada host cell tu, pada bacterial cell. One is the bacteria that does not take any plasmid. Second, bacteria that take up non-recombinant plasmid. Yang ketiga adalah bakteria yang take up recombinant plasmid. So macam mana kita nak tahu sekarang ni bakteria mana yang mana? Itu keperluannya screening. Screening ni kita gunakan yang selectable marker gene tu tadi. Okay, so kegunaan ampicillin resistant gene adalah untuk screen bakteria yang take up plasmid dengan bakteria yang that does not take up any plasmid. So dekat medium ni nanti awak campurkan dengan ampicillin resistant gene. Okay, so bakteria yang tak ambil any plasmid akan mati. And ataupun selectable marker, any bacteria resistant medium in the ampicillin medium kan. Kalau bakteria yang take up plasmid, tak kisahlah dia non-recombinant ataupun recombinant, so bakteria ni boleh hidup. You can live on. Okay, itu fungsi the first selectable marker is to screen bakteria mana yang ada plasmid yang bakteria mana yang ada non-recombinant plasmid, bakteria mana yang ada recombinant plasmid. Itu fungsi reporter gene. Okay, dalam note tapi ada dalam substance dinamakan sebagai gal X. Saya tulang. Like that. Enzyme. Putih, dua koloni. Sama ada warna putih ataupun biru. Cuma saya tak ingat mana satu koloni. Siapa-siapa boleh cari sekejap Okay, nampak saya punya presentation ni? Okay, sorry. Berapa kali terputus. Okay. Yang lain boleh dengar ya? Saya nak sambung balik ni.
Boleh, Madam.
Okay, sorry. Alright. Okay. Okay, so tadi saya nak cari blue and white colony tu kan? Okay, sekarang ni bila awak mixkan bakteria dengan plasmid, so ada 3 possibility saya cakap tadi kan? So sama ada bakteria yang tak ambil any plasmid, bakteria yang ambil non-recombinant plasmid dengan bakteria yang ambil recombinant plasmid. Okay, ada 3 possibility. So sekarang ni awak nak screen, awak nak pilih mana satu bakteria yang ambil plasmid. First awak nak kena determine sama ada bakteria yang ambil plasmid ke tak. So macam mana awak nak tahu, awak kena itu kegunaan marker gene ampicillin resistant gene. So bakteria ni medium dia awak letak dengan antibiotik ampicillin. So bakteria yang mati adalah bakteria yang tak ambil any plasmid sebab dia tak ada ampicillin resistant gene. Okay, so itu screening yang pertama. Okay, you screen the bacteria that has taken up plasmid or not. By adding any bacteria that does in bacteria sama ada dia take up non-recombinant plasmid atau dia take up recombinant plasmid. Itu adalah kegunaan gene yang kedua iaitu reporter gene. In this case, kalau kita tengok kepada like Z gene, pada plasmid yang ada like Z gene, dia akan menghasilkan enzyme beta-galactosidase. Kita dah tengok hari tu masa kita tengok the function of beta-galactosidase is to hydrolyze lactose kan into glucose and galactose. Tapi dalam dalam eksperimen ni, we do not use lactose. We use another substance yang kita namakan dia sebagai GalX. So maksudnya dalam medium yang ada bakteria lagi, awak campurkan dengan GalX substance ni. Okay. So pada bakteria yang ada plasmid, obviously dia akan ada like ZG ni. Sepatutnya kalau like ZG ni tak diganggu, dia boleh menghasilkan beta galactosidase. Dekat like ZG ni akan ada restriction site. Okay. So restriction site ni akan dipotong oleh restriction enzyme dan dia akan insert dengan foreign DNA. Okay. So maksudnya plasmid yang akan jadi recombinant plasmid, so sekarang ni dia dah tak boleh dah nak hasilkan beta galactosidase. Okay. Any bacteria that take up recombinant plasmid will not be able to produce beta galactosidase anymore because the lysozyme gene now is not functional. Why? Because the lysozyme gene ni has been cut. Okay, sebab apa cut? Sebab dalam lysozyme gene ni ada restriction site. Okay, so this is where you cut the plasmid and then you insert the foreign DNA and become recombinant plasmid. So any bacteria that take up recombinant plasmid will not be able to produce beta galactosidase and will not be able to hydrolyze gal X. Okay, so bacteria yang take up recombinant plasmid ni dia akan menjadi koloni yang berwarna putih, white colony. Okay, so kalau bakteria yang take up non-recombinant plasmid, maksudnya recombinant plasmid dia bercantum, elok, tak ada insertion of foreign DNA. So like zat gene ni dia boleh berfungsi seperti biasa dan menghasilkan beta galactosidase dan boleh hydrolysekan gal X ni dan menjadi, bila dah hydrolyse gal X ni dia akan jadi substance yang berwarna biru. Okay, so this produce the blue colony. So sekarang ni order skin, bakteria mana yang ambil recombinant plasmid, bakteria yang mana ambil recombinant plasmid. So sekarang ni awak nak ambil koloni yang warna apa? Kelas, awak nak ambil koloni yang warna apa yang ada that may contain gene of interest? Koloni biru ke koloni putih? Anak-anak sekalian, nak ambil koloni putih ke biru? No idea. Ke tak dengar saya cakap, saya terputus lagi ke apa ni? Nampak tu
No
dekat
idea.
Ha?
No idea.
No idea. Awak tengok dekat tools yang nombor 6 ni, marker gene kan. Fungsi like Z gene ni adalah untuk menghasilkan beta galactosidase untuk blue white screening. Kadang not ditulis kan. So blue white screening ni apa? Dalam dia, bila awak culture bakteria tu, dia akan menghasilkan dua koloni yang berwarna. Satu koloni warna putih, satu koloni warna biru. Koloni putih terhasil disebabkan oleh bakteria yang ambil recombinant plasmid. Catat koloni yang berwarna putih adalah bakteria yang ambil recombinant plasmid. Sebab apa dia jadi koloni yang warna putih? Sebab like Z gene dia sekarang ni dah tak berfungsi. Dia dah tak boleh nak menghasilkan beta galactosidase. Okay. Bila dia tak boleh nak menghasilkan beta galactosidase, dia tak boleh nak hydrolysekan substance gal X tu. Okay, so dia maintain as warna putih. Tapi pada bakteria yang ambil non-recombinant plasmid, okay, non-recombinant plasmid maksudnya plasmid dia still utuh, the like Z gene still function and can produce enzyme beta galactosidase. So bila awak letak dia dalam medium yang ada gal X, bakteria-bakteria yang ambil non-recombinant plasmid boleh hydrolysekan, convert the gal X substance into a blue substance. So this will cause the bacteria that take up non-recombinant plasmid to form blue colonies. So now, which colony do you want? The blue or the white?
Blue.
Blue.
Kenapa blue? Blue ke white? Awak nak bakteria yang ada non-recombinant plasmid ke? Awak nak bakteria yang ada recombinant plasmid?
Yang ada.
Dan recombinant?
Dari
Dan recombinant? Ya, awak sisi lah kerja awak.
Recombinant. Recombinant, madam.
Ambil white.
You want the bacteria with the recombinant plasmid, the foreign DNA. Foreign DNA you insert dalam tengah-tengah gene yang ada like Z gene ni. Baik semula lah. Ini saya draw balik semula. Dish. Dan ni awak letak adalah antibiotik. Bakteria yang ini plasmid hanya yang ini. So bakteria ni mati. Tak ada ambil any plasmid. Bakteria yang kedua adalah bakteria yang ambil recombinant plasmid. Dan recombinant plasmid apa dia? Plasmid bila awak dah potong dengan restriction enzyme, cantum balik. Dia tak ambil AD foreign DNA, dia bercantum balik. Ini adalah bakteria yang macam ni. Dia ambil non-recombinant plasmid. Stability yang ketiga adalah yang saya buat warna oren ni lah. Okay. Bakteria yang ambil recombinant. So ini adalah bakteria yang mana plasmid dia ada insertion of foreign DNA. Ini adalah contoh bakteria yang macam ni. Draw ni. So these are the bacteria with the recombinant plasmid. Ada 3 possibility. Fungsi marker gene ni. So marker gene yang pertama. Resistant gene. Insulin resistant gene akan mematikan any bacteria yang does not take up any. Bacteria ambil plasmid tak kisahlah recombinant ke, dia boleh survive. Boleh survive. Sebab apa? Dia ada plasmid. Sekarang ni awak tambah pula dekat dalam medium tu dengan substance yang kita namakan dia sebagai LX. Substance ni dia molekul dia lebih kurang sama dengan lactose. So itu kegunaan reporter gene. In this case, kita nak concentrate pada reporter gene yang ada Z. Pada DNA plasmid ni tadi, DNA plasmid mana awak insertkan DNA, kawasan yang awak cut plasmid tu kita namakan sebagai restriction site. Initially, belum. Ini sebelum dia jadi recombinant DNA macam tu, plasmid dia macam ni. Ini ada gene untuk Z gene. Region yang ada Z gene ni, kawasan ni adalah kawasan restriction site. Restriction site tu adalah site yang mana restriction enzyme open the plasmid for the foreign DNA to be inserted and produces recombinant plasmid. So bila awak dah cut open the plasmid, you cut open the region between the like Z gene. So now like Z gene is not functional. Not functional. Not produce enzyme galactosidase. So the bakteria yang ambil recombinant plasmid, like Z gene dia masih berfungsi, beta galactosidase can function. Tunjukkan dia kat ni lah. Z gene fungsi boleh menghasilkan beta galactosidase dan akan convertkan LX di blue substance. So, this bacteria will become blue colonies. Blue colony. Okay, ini akan jadi blue colony. Jadi blue colony. Akan jadi blue colony. Pada bakteria yang ambil pada bakteria yang ambil recombinant plasmid, the like Z gene cannot function, cannot produce beta galactosidase, cannot hydrolyze GalX, cannot produce blue substance. So this become the white colonies. So saya indicate dengan warna yellow ni. So this is the white colonies. So awak nak bakteria ada recombinant plasmid. Obviously, you want the white colonies. Because the white colonies will have the of interest. Blue colony will not have the gene of interest sebab dia dah bercantum balik semula kat sini ni. Faham ke?
Faham, Madam. Faham.
Kita ambilkan a few slide dekat sini kejap. Yang stage keempat, amplification of target gene by host cell and selection. So the host cells are cultured in a medium containing ampicillin, which is antibiotic. Host cell with recombinant DNA reproduce through repeated cell division. Sebab apa? Cell ni boleh survive sebab dia ada plasmid. So this is as you can see from the diagram here. Okay, so these are the bacteria. Kejap. So these are the bacteria that take up the recombinant plasmid. Recombinant plasmid and also ada bacteria yang ambil non-recombinant plasmid. So you grow in the medium containing antibiotic. Bakteria yang tak ada plasmid dia akan mati. Yang ni, bakteria yang ni. Tapi bakteria yang take up recombinant plasmid dalam ampicillin dia boleh survive. For ampicillin resistant gene, ni for the recombinant plasmid. Of gene cloning, production of insulin. So here is the human cell. insulin. So you extract out the DNA, you cut with restriction enzyme, and then you produce DNA fragment that may contain gene of interest that, for example, produces or code for insulin. So this is the bacterial cell. You extract out the plasmid, and then you cut open using the same restriction enzyme. So you mix together the cut open plasmid and the foreign DNA together to produce the recombinant DNA. Then you mix balik semula dengan bacterial cell. So ada bakteria yang ambil plasmid, ada bakteria yang nak ambil. Yang tak ambil mati, yang ambil hidup. So the bacteria that survive will undergo cell division and replicating. Itu yang kita namakan dia sebagai cloning. Faham? Cloning. So as the bacteria divides, produces many cells, it also replicates the plasmid that contains the gene of interest. So kegunaan dia sama ada dua. Ada you want the gene, you isolate balik semula gene insulin tu dengan kuantiti yang banyak sebab bakteria tu dah pembahis dengan banyak, ataupun extract out the product the insulin produced by the bacterial cell, because the bacterial cell is not has the gene insulin gene, it can also produce the insulin. Okay, so kita stop sampai sini dulu. Ada soalan ke nak tanya? Any questions? Two slide.
Faham.
Kalau tak ada soalan, make sure you all tengok balik notes. Okay. Next week kita sambung. Sepatutnya next week kita masuk homeostasis. So kita macam terlambat. Jadi maybe minggu depan buat extra class lah. Okay, sebab saya nak habiskan chapter ni. Jadi saya akan ambil time session. Kalau tak ada soalan, make sure you all have signed. Okay. Make sure you sign. You can check lah the Google Meet. Sure sign, lepas ni tak nak ada yang saya tak terlupa. Ke depan kita ada quiz genetik hari Rabu.